Assalamu alaikum everyone. Today's topic of discussion is antibiotic resistance, and we will talk about what is antibiotic resistance, how does it develops, and what are the uh, two basic mechanisms of development of antibiotic resistance, that is genetic basis as well as non-genetic basis. So let's start our topic antibiotic resistance of a bacteria to the effects of the antibacterial agent. How does bacteria develop resistance? For this, we should know the mechanisms of actions of antibiotic drugs. So first of all, we have the inhibition of cell wall synthesis. Cell wall is actually made up of peptidoglycan, and peptidoglycan have chains and they have cross linkages. Then we have inhibition of protein synthesis. Protein synthesis involves 30S subunit, 50S and transfer RNA. Then inhibition of nucleic acid synthesis. Nucleic acids include nucleotide, DNA, and RNA. So, nucleotides include D, DNA nucleotides and RNA nucleotides. Then we have disruption of cell membrane and other mechanisms. Other mechanisms have mycolic acid synthesis inhibition, DNA damage, and fatty acid synthesis inhibition. So, how does resistance develops? Inactivation of antibiotic drug, that is cleavage by beta lactamase enzyme. Second mechanism is modification of target of drug in bacteria by bacteria itself. Then modification in penicillin binding protein or receptor which affects penicillin. Then modification or mutation in 30S subunit, drugs acting through protein synthesis inhibition are affected. Then we have mutation in DNA gyrase. DNA gyrase acts through mutations in DNA synthesis. Then mutations in RNA polymerase, that affects the drugs which act through RNA synthesis inhibition. Then mutations in peptidoglycan, lactate replaces alanine, and this affects the drugs which act through cell wall inhibition. So first mechanism inhibits the action of inhibition of cell wall synthesis. Second one affects the inhibition of protein synthesis. Third one affects the inhibition of DNA synthesis, and so on. Now we'll discuss two basic types of antibiotic resistance, that is genetic and non-genetic. In genetic we have chromosomal mutations, plasmids and transposons. Chromosomal mutations include mutations of the genes which are involved in the synthesis of antibiotic targets. Then we have plasmids. Plasmids may we have antibiotic or drug resistant genes and transfer genes. So, antibiotic or drug resistant genes are present on R factors, and they may be more than one or multiple antibiotic resistance genes. As you can see in this plasmid, which has genes for gentamicin resistance, penicillin resistance, erythromycin resistance, and vancomycin resistance. So, hence resistant to all of these antibiotics. And plasmids have transfer genes, and plasmids can be transmitted from one cell to another, and they can be replicated independently, and transmission occurs through conjugation by sex pili. Then third mechanism for the genetic basis of antibiotic resistance is through transposons. Transposons are the moving or the jumping parts or segments of the DNA which can move from one part of the DNA to the other, and they have three type of genes for drug resistance, which include transposase, which is involved in the regeneration of the transposon, as telomerase is involved in the synthesis of telomere. Then we have the repressor gene, which represses the synthesis of transposase. And lastly, we have drug resistant genes, which are actually involved in transposon related drug resistance. I've attached here this table for the mechanisms of actions of antibiotic drugs with examples of each category of antibiotics. And here is what our actual topic was about. This is the mechanisms of antibiotic resistance, and drugs affected in each case are also listed. Thank you so much for watching the video till end, and please share the video, like the video, and press the subscribe button and the bell icon too.